All right, let's move it along to our next. It seems like we're going back and forth this morning. We're going eyes oncology, eyes oncology, eyes oncology. We're going back to oncology here with our next presentation. Salarius Pharmaceuticals. David Arthur is the CEO. I see him here, guys. Let's bring David on. David, there he is. How are we doing? Good morning.
Good morning, Spencer. Thanks for having us, and glad that you're getting lots of oncology companies in here today.
Yes. Well. Yeah. The day is young yet. The day is young. We have the whole rest of the day. We got you guys. We got tomorrow. But yeah, we're talking oncology. We're talking everything here at the Benzinga Small Cap Healthcare Conference. So that being said, I'm going to bring on your slides. There they are. I'm going to step backstage and I'm going to give you the floor, sir, to tell us all about it.
All right. Well, Spencer, thank you for the introduction, and I'd like to welcome everyone who's taking the time to learn a little bit more about Salarius. Let's jump in. And as Spencer said, we are an oncology company. And let me hit the highlights, and then we can get into a little bit of the detail. We are a clinical stage biotechnology company, publicly traded on NASDAQ under the symbol of SLRX. You can see that scrolling along the bottom of the screen here. And we are currently targeting drugs that deal with dysregulation. And the best way to describe that is, relating it to cooking and following a recipe in the kitchen. If you follow the recipe correctly and make no mistakes, then you end up with exactly what you were hoping for. Unfortunately, sometimes we misread the recipe. We add pepper instead of salt. We use half a cup instead of a teaspoon, or perhaps we use the wrong temperature in the oven, or it's in the oven for too long or too short a period of time. We misread the recipe. That is exactly what can happen in the human body. And when that happens, you get dysregulation, and the body can inadvertently create these proteins that cause cancer, and that's called epigenetics. So really just think about it as the body misreading healthy genes. Our lead drug is an oral tablet that targets an enzyme that the body we believe is critical for these oncoproteins that have been created because the body misreads healthy genes. This enzyme is critical in those proteins telling the body to start and grow and progress cancer. And what we found is there's a number of cancers that we believe we can positively influence, meaning we can show anti-cancer activity, and there's a group of sarcomas, and we've also recently moved into hematologic or blood cancer. So we have a speed to market, huge unmet need in some sarcomas. We have very positive interactions so far with the FDA. We've received rare pediatric disease designation, which sets us up for a voucher. It's worth $100 to $125 million when we have approval for Ewing sarcoma, one of the sarcomas we're studying. Orphan drug designation, fast track approval. So this all represents speed to market. Hematologic cancers represent one of our expand the market opportunities. Now, we are in the strongest financial position we've ever been in. In fact, earlier this week, we announced the receipt of, I believe, $2.7 million from the state of Texas. In June of this year, we reported cash on hand of $33 million, and we still have $2.1 million remaining in an award that we received from the state of Texas back in 2016. So wrapping up where we are, speed to market, we're in the clinic, active clinical studies, recruiting, I believe, 5 different types of patients in treating them with single agent and 2 different combination therapies. Speed to market with these very, very well identified sarcomas with high unmet need, and much larger markets like blood cancer. And we'll talk a little bit more about that in a moment. So where are we specifically? Well, we have a study ongoing, phase 2, treating up to 60 patients with our recommended phase 2 dose. And you can see 3 different types of sarcomas. One in Ewing sarcoma, devastating disease in combination with topotecan cyclophosphamide. And it's important to note that in cell lines, Ewing cell lines, or Ewing cell line, we have shown synergy with Seclidemstat and TC therapy, which means 1 + 1 is much more than 2 in attacking these cancers. In myxoid liposarcoma and FET rearranged sarcomas, the single agent therapy, we've had some very, very interesting and what we think is supporting data coming out of an earlier clinical trial that tells us, we may really be able to move a needle in these hard to treat sarcomas. I mentioned hematologic cancers, again, being treated with combination therapy, azacitidine, Seclidemstat, and Honor Health in Arizona has announced a study in select gynecological cancers where we're identifying mutations which may make patients more susceptible to LSD1 inhibition therapy, where we inhibit that enzyme. And in this particular study, we're treating in combination with what's called a checkpoint inhibitor, because what we believe is that Seclidemstat can unmask these hidden tumors and allow checkpoint inhibitors like Keytruda to now attack these cancers. So this is a very robust clinical program that we are really looking forward to being able to talk about later this year and next year when we have patients enrolled and having been treated for a while, and really be able to talk about the difference we can make in patients' lives. And as you would imagine, for a company focusing on LSD1 inhibition, we have a second generation program, early discovery, ongoing at the same time. So let's dig into the data a little bit. Now I'm going to focus on the right-hand side of this slide. The left-hand side, the left-hand component gives us a very, very scientific description of how epigenetic drugs work. The right-hand side is a little more straightforward version and story. As I mentioned, the body can misread genes, and that creates oncoproteins that will tell the body to begin growing, start to grow and progress cancer. And they do that by dysregulation, which turns tumor growth genes on and turns off tumor suppressor genes. If we can correct that, and we've shown it in cell lines and in animals, you correct that dysregulation and you create balance, and you cut off the body's ability to send these cancer-generating communication signals. It's a very well-validated space, both epigenetics and the pursuit of LSD1 inhibition. And when you look at the right-hand side of this slide, and you see some of these names up here, you begin to see that there are a lot of really well-recognized companies going after this type of approach, inhibiting the LSD1 enzyme. And when you look at the left-hand side of the slide, you can see examples of very successful companies that have pursued epigenetics, although in different types of areas. I draw your attention to Imago, down on the lower right-hand side of the slide, that recently went public as a company with a lead compound as an LSD1 inhibitor, and they are currently trading at about a $750 million market cap, which gives you an idea of the potential of being able to demonstrate anti-tumor activity with these types of drugs. We believe we're about 12, 15 months behind Imago. They're about 12, 15 months ahead of us in enrolling in their phase 2 study, and we've just begun. So I think there's a lot of validation out there for the approach that we're taking. How does the drug work? Well, any LSD1 inhibitor is going to inhibit the enzyme. And so you can see on the far left-hand slide, everybody inhibits the enzyme. But where we, Salarius and our LSD1 inhibitor, provides real differentiation is we have much broader scaffolding properties and inhibition than the other LSD1 enzymes. And this is important because it gives us a broader range of activity. And by interrupting the protein-protein interactions, we deliver that differential activity and we reduce LSD1 expression, which has been shown to be associated with higher mortality rates in a number of cancers, such as prostate, breast, and ovarian. Because we are a reversible inhibitor, and two of the other clinical medicines pursuing LSD1 inhibition are irreversible, we believe we have a favorable toxicity profile. So, in summary, everybody does the enzymatic inhibition, but Seclidemstat, we believe, has much stronger and broader scaffolding inhibition, which leads us to having better differential activity and reducing LSD1 expression. So what about the markets we're pursuing, and why should we be excited? Well, first of all, these are areas with huge unmet need, and one of our lead indications, Ewing sarcoma, is a devastating illness, bone and soft tissue cancer that affects kids. The average age of diagnosis is about 15 years, and really the only treatments available are adult chemotherapy, radiations, and surgeries. It works in about 50 to 60% of the patients, but you can imagine the long-term complications of treating kids with these types of therapies. And unfortunately, where it doesn't work, in about 40% of the patients, you have about an 80%, 70 to 90% 5-year mortality rate. It's just devastating in a group of people and patients that have their whole life ahead of them. Well, as I mentioned, we are pursuing a non-toxic or less toxic oral treatment. We've had great interactions with the FDA. We're set up for a priority review voucher. And if we can replicate what we're seeing in animal studies, we really think we're going to move the needle on patient care. So let's take a look at what we've seen already in preclinical studies. So on the left-hand panel, we work by inhibiting the LSD1 enzyme. You can see that highlighted here in purple with the little red arrows. And what happens is these oncoproteins that have been incorrectly created because the body misread the healthy genes, their binding and use of the LSD1 enzyme is interrupted. You look at the middle panel and you can see what this looks like from a genetic and a transcriptional profile perspective. On the left-hand side, all these red tumor growth genes are incorrectly turned on, while all these blue tumor suppressive genes are incorrectly turned off. And these are Ewing's sarcoma cell lines. When you look on the right-hand side of the center panel, you see the same cell lines treated with an analog of our lead product, Seclidemstat, and you begin to see a complete reversal of that transcriptional profile. And when you look on the right-hand panel, you see that when you put this drug in animal models where you're growing tumors, you can see in the lower lines on both of these graphs that the tumors, without that information and that messaging to continue to grow, they slow down, the tumors plateau, they begin to melt away. This is what we're hoping to achieve in patients. It's what we're doing now. And to supercharge this single agent therapy, as I mentioned, we're treating patients with combination therapy where we have synergistic 1 + 1 = more than 2 anti-cancer activity. We've already treated some patients, and a picture is worth 1,000 words. These slides are available on our website. I encourage you to look at them in detail, but what I want to point out is right here in the middle. What you see in this is a scan of a patient, and what you see is this, in the middle right here, is a large tumor. When you look on the right-hand side with the red, that tumor is gone. This patient was treated with Seclidemstat for 6 months, and they had continuous target lesion reduction. Target lesions are the lesions that doctors chose to measure. Now, unfortunately, the patient did have progressive disease in another area of their body, but this is exactly what we were looking for early on in our dose escalation study. So this is the positive data that really caused us to want to accelerate our clinical program. But as I mentioned, we're in more than Ewing sarcoma. And one of the reasons is shared biology. And when you look at this bottom left quartile of the panel, you can see the relationship between Ewing sarcoma and these other sarcomas. And because of what we saw in the Ewing study and what I'm going to show you in a moment, we expanded our sarcoma study to include these additional sarcomas, which we also believe could represent accelerated approval opportunities. Now, quick snapshot, right-hand side of the slide. All of these descending lines represent cell lines and anti-cancer activity or cell viability is demonstrated by our drug, 2577. Why are we excited? Well, in a clinical trial that we have now completed, we had 3 patients with these sarcomas enrolled in the study. And I'm drawing your attention to the bottom graphic. And you look at the far side and you see time to progression. Well, these patients had 4.3 months, 7.2 months, and 12 months and counting. They're still on drug. And that was how long it took them to progress using single agent therapy. And previously, their best response had just been stable disease. Well, if you look at the literature would tell you that for advanced soft tissue sarcomas, about 3 to 3.5 months is very typical for treatment at this stage of illness. And 100% of the patients that we treated with single agent therapy exceeded that number. When we saw this data, the first thing we did was add these patient types to our ongoing sarcoma study. If we can replicate this, we believe this is a registerable indication. That's all speed to market. When we look at market expansion, we're beginning to look at how, as I mentioned earlier, how can we identify patients that have mutations that will make them more susceptible to LSD1 inhibition therapy. 2, we are looking at the ability to use our drug in combination with checkpoint inhibitors, which are about a $20 billion a year market. And they only work in about 40% of the patients because many times the body can't see the tumors. Well, we can unmask those tumors, we believe, and allow these checkpoint inhibitors to work more effectively. And then finally, as I mentioned, there's been some great information on LSD1 inhibitors in hematologic cancers. MD Anderson felt the same way and has launched a study using Seclidemstat in 2 types of cancer where about a third of the patients advance to have acute myeloid leukemia. And it's important to note that according to the American Cancer Society, acute myeloid leukemia accounted for about 20,000 new cases of cancer in 2020 alone. So we have a number of activities that are all pursuing much, much larger indications. So let's wrap it up and get to some Q&A. What's the opportunity with Salarius? Well, speed to market. We're in a great sarcoma trial treating 3 different patient groups. One of those groups is in combination therapy where we have shown synergy data, which means 1 + 1 = more than 2. We have pre-approval for a voucher that I'm happy to talk about in Q&A, that if we are approved for Ewing's, we believe we will receive this voucher and we can monetize it. We're looking at a very nice global sales line in the sarcoma area. But an even bigger opportunity is the expansion into larger markets. And as you can see, MD Anderson has initiated that study. We have Honor Health, who has announced their study. They just haven't begun enrollment. And then, as I mentioned previously, we are looking to continue to identify how to select patients to not only enrich our clinical trials, but also provide us with commercial targeting for patients that will best respond to therapy. So with that, this is the summary of Salarius. We are publicly traded on NASDAQ. I think for individuals who are building a biotechnology portfolio, Salarius is something you should take a real look at. With that, I'll wrap up the presentation and open the floor to questions.
Thank you, David. I didn't think we were going to have time, but I do want to squeeze one question in from our chat, which is there a specific kind of cancer that you're most effective against here? Or
well, excellent question. I believe we will be effective, more effective in certain cancers rather than others. And that's one of the reasons we're doing our clinical trials the way we are. We have a broad variety of cancers that we've identified where we already think we're going to be successful. And we're beginning to explore the best way to treat patients, either as single agent or in combination. And so when you look at the clinical data that we have, you can see that we are making a difference in both Ewing sarcoma and these other FET rearranged sarcomas. And right now, I would say that is where we're focusing our efforts because of the tremendous unmet need and the potential for accelerated registration.
All right, David Arthur is the CEO of Salarius Pharmaceuticals. David, thank you so much for joining the Benzinga Small Cap Healthcare Conference. Have a good rest of your day.